Hey everybody, let's talk about these summative slides for the nervous system. So the first thing you're going to want to know is what is the major function and the major structures of the nervous system. The major components in the nervous system are the central and peripheral nervous system. Central nervous system is made of the brain and spinal cord, and the peripheral nervous system is made of all of the nerves that are not in the brain or spinal cord. So all of those neurons that are sensory or motor neurons. What are the purposes of the nervous system? The purposes of the nervous system are to receive sensory information, process that information, and then generate an appropriate response. So receive information, process it, and then respond. That's the purpose of your nervous system. The nervous system is divided into two major divisions, like we talked about earlier, central nervous system and peripheral nervous system. We should also know that the peripheral nervous system is divided into two branches, the sensory and the motor nerves. So your sensory nerves are going to be sending information to your central nervous system, and then your motor nerves are going to be taking information from your central nervous system and directions from it and sending that information to your muscles and to the body. The motor neurons are branched into two further subcategories, somatic and autonomic. So all of your motor neurons are either somatic or autonomic. Somatic means you have voluntary control over them, and that controls your skeletal muscle movements like this. Your autonomic motor neurons have control over the automatic functions of your body, things like digestion, peristalsis, the contraction of your smooth muscles. All of those automatic signals of your autonomic nervous system, so your motor nerves, the autonomic motor nerves, are branched into two categories, sympathetic and parasympathetic. Your sympathetic nervous system is fight or flight, and your parasympathetic is rest and digest. So very excitatory, heart rate increases, breathing increases, pupils dilate, parasympathetic, rest and digest. That is regulating things like digestion, peristalsis, vasoconstriction, vasodilation, that kind of stuff. Cool, moving on. Your nervous system is made up of neurons. Those are the primary cell type of the nervous system, right? Your neurons, they're the ones that are going to be transmitting information. You should know the anatomy of a neuron. Up here we have the dendrites, where they will receive a stimulus. Then there's the cell body, which includes the nucleus. Then we have this spot here called the axon hillock, where the axon branches off of. This long extending projection here is the axon. And we can see that it is covered in these sections of insulation called Schwann cells. You should know that this is a myelin sheath, right? So this is myelinated axon, which increases the speed of an action potential or a speed of the signal down the axon. Then we also have the axon terminals down here. That's where there's going to be a synapse. That is where neurotransmitters are released and a signal is sent to the next neuron or to the muscle. These little gaps between the Schwann cells are called the nodes of Ranvier. If you don't know that one specifically on the test, that's okay. Okay, moving on. Let's talk about how does an action potential work? Well, first of all, a stimulus is received at the dendrites, right? That stimulus is going to depolarize this membrane a little bit. If it reaches threshold, then it'll depolarize all the way and it'll hyperpolarize, and that is what we call an action potential, and then it hyperpolarizes and gets back up to that resting membrane potential. Let's walk through that step by step here. Sorry, the screen is a little bit funky. Let's go like this. Can I move this? Perfect. No, not right there. Oh, that'll work. Okay, step one. The sodium-potassium pump moves potassium in and sodium out of the axon, creating a resting membrane potential of negative 70 millivolts. What does this mean? This means that at rest, your membrane potential, hold on, I'm going to pause this. Let's look at another slide. Hello, jump cut. On the actual axon itself, let's see, this is the outside of the axon, this is the inside of the axon, this is the outside of the axon. You have all these proteins embedded in the membrane of that neuron. So you have this one here, which is the sodium-potassium pump. I'm going to move myself over here. The sodium-potassium pump is pumping sodium out of the cell and potassium into the cell. Both of those are positive molecules, but we're pumping more sodium out of the cell than we are pumping potassium in. So over time, this creates a negative gradient. So the inside of the cell is going to be negative with respect to the outside of the cell. And we measure that in millivolts, right? It is negative 70 millivolts, right? So it is 70 millivolts less positive than the outside. Okay, that is our resting membrane potential, and it is maintained by these sodium-potassium pumps. So that's our neuron at rest. Okay, then at the dendrites over here, we get a signal that is going to slightly depolarize the membrane. We get a little bit of a signal. Now, if that's all we get, then the sodium potassium pumps are going to continue to bring it right back down to membrane potential. But once we reach this threshold, if we get enough of an excitatory stimulus to reach this threshold, then that is automatically going to trigger these yellow proteins, which are called voltage-gated sodium channels. Those voltage-gated sodium channels automatically open at negative 55 millivolts. Once that happens, then sodium can flood into the cell because you can imagine there's a lot of sodium outside, there's not a lot of sodium inside. The sodium wants to flood inside, especially because it's super negative in here with respect to the outside. So you have a chemiosmotic gradient. Those sodium ions will flood in, which will cause the membrane potential to shoot up. That is called depolarization. That is depolarization. And we go from negative 55 all the way up to around positive 40. Now, at positive 40, those voltage-gated sodium channels close, and these voltage-gated potassium channels open. So now that these voltage-gated potassium channels open, those potassium ions that were in high concentration inside the cell, they flood out of the cell. And remember, potassium is also positive. So since all those positive ions are leaving the cell, then the membrane potential crashes all the way back down. This is called repolarization. Now, after repolarization, it actually hyperpolarizes a little bit. So we actually shoot past the resting potential, but the sodium potassium pump is going to continue to pump, and it will bring us back up to resting membrane potential. That is an action potential. How does that actually travel down the cell? Well, if we take a look at the way that this would work, step one, we receive a stimulus. If that step gives us enough as a signal to reach threshold, then these automatically open, and then sodium floods in. And now that sodium is flooding in over here, this part of the cell is a little bit more positive, which is going to trigger this sodium channel. And then it's going to flood in here, and that's going to trigger this sodium channel. So it's kind of like a chain reaction or domino effect, where as soon as it becomes positive on this side of the axon, then it's going to be a little bit more positive over here, which will open up this channel, and then that will continue all the way down the axon. And then you have all the way down the axon this wave where the axon is becoming positive. But directly behind that wave, you have the potassium channels opening and potassium flooding out, so it becomes negative again. So you just have this little blip that is positive that travels all the way down the axon, down to the axon terminals. And that movement of charge, right, of that little positive movement, is electricity. That is what electricity is. Electricity is a movement of charge. And that travels down to the axon terminal, where we have a synapse. Now, a synapse looks like this. So if we're taking I'm just going to hang out over here, our neuron where we got our stimulus the first time, and then we have that action potential travels all the way down the axon and is released here at the axon terminal. Let's take a look. Let's see that this was the axon of another neuron, and it is sending a signal to this neuron here at these dendrites. So this little box right here is where we're zooming in. So this would be part of the axon here, and this part in yellow would be the postsynaptic neuron, but it would be the dendrites. Okay, so this is how, when that action potential reaches the end of the neuron, it arrives in the axon terminal. That positive charge, once it arrives, it triggers the release of neurotransmitters. We have these synaptic vesicles that are full of neurotransmitters, and they fuse with this membrane, just kind of releasing them as they go. Those neurotransmitters then bind with ligand-gated channels, so not voltage-gated channels, ligand-gated channels. We could also say neurotransmitter-gated channels. So as soon as that neurotransmitter touches this channel, it'll open, allowing ions to flood in, which will slightly depolarize that neuron. And if that's enough of a signal, it'll actually trigger another action potential, which will travel down its axon. So what do we need to know about a synapse? A synapse is where the axon terminals of one neuron reach the dendrites of another neuron, or technically the muscles. And when an action potential travels down that presynaptic neuron, the action potential reaches the axon terminal, and it signals the release of neurotransmitters. Those neurotransmitters bind to the dendrites of the next axon, which then depolarizes that cell, in order to trigger another action potential. So this first axon, this first neuron is sending a signal to the next neuron. Now, it is important to note that the effect of that neurotransmitter could either be excitatory or inhibitory. So it could make it more likely that an action potential is going to happen in that cell, or it could be less likely that an action potential is going to happen in that cell. And there are all sorts of different neurotransmitters like dopamine, serotonin, GABA, glutamate, and they all have different effects. Watch this video if it makes more sense there. Okay, next thing we need to know, we need to know the major divisions of the brain. So your brain is made of 3 major parts. Ooh, let's go like this. 3 major parts. We have the brainstem, the cerebellum, and the cerebrum. Here's what you need to know. The brainstem is made of the midbrain, the pons, and the medulla oblongata, and it is in control of your involuntary movements. So stuff like peristalsis, your smooth muscle contraction. Those signals originate here. Then you have your cerebellum, which is a little bit darker and sits kind of in the back of your skull. Cerebellum is in control of your voluntary movement. So the signals that originate from your brain that say, hey, I'm going to go move muscles, they come from here. So involuntary movement is controlled by the brainstem, and voluntary movement is generally controlled by the cerebellum. Then we have the cerebrum, which is this whole wrinkly part that we like to think of as the brain. The cerebrum is in charge of your higher level functions like cognition, like memory, like learning, emotion. That's where you are. That's where your personality is. Okay, this outside portion is called the cortex, and it's full with all these little grooves. What you need to know is that you have 2 hemispheres, the left and the right hemisphere. They're connected via this little arch shape right here called the corpus callosum. Whoop. And there are 4 lobes, the frontal, temporal, parietal, and occipital lobe. So occipital lobe, frontal, the parietal, and the temporal lobe. And then there are a lot of subcortical structures. So that was all the cortex. It's made of gray matter, which is mostly the cell bodies of the neurons and unmyelinated axons. And then the white matter, which would be the inside part, is made of myelinated axons. It's like the wires that are connecting them. Did I forget anything there? I think that's it. You don't need to know any of the subcortical structures specifically, except for the corpus callosum, which is what connects the left and the right hemispheres of the brain so it can communicate with itself. Okay, if you have any questions, send me an email. I'd be happy to help out. I hope this helps. I would wish you good luck on the test, but you don't need it.